Look at a standard agricultural field, like this massive expanse of jute. For thousands of years, scaling up production simply meant planting more seeds and hoping for favorable weather. But to truly scale biological output today, we have to bypass the macroscopic level entirely. We dive past the leaf surface, into the cellular structure, right down to the DNA double helix inside the nucleus. This is the domain of biotechnology. Hungarian engineer Karl Erecki coined the term in 1919 by combining biology with technology. It is the targeted application of technological processes to biological systems. We are modifying living cells to manufacture products. Modern biotechnology rests on two massive pillars. First, replicating identical, high-quality organisms at scale, known as tissue culture. And second, altering their intrinsic capabilities, known as genetic engineering. These two methods, copying the whole and rewriting the part, allow for the deliberate manipulation of biological systems. The foundation of tissue culture relies on a biological axiom called totipotency. This is the intrinsic ability of a single, isolated plant stem cell to contain the complete genetic blueprint required to regenerate an entire, fully functioning organism. The protocol begins by extracting an explant, a tiny fragment of target tissue taken directly from a healthy donor plant. The explant is placed into a nutrient medium. It uses agar to create a semi-solid base, sucrose for energy, and specific phytohormones calibrated to force cellular growth. But there is a massive biological bottleneck. This environment is perfectly primed for growth, which means ambient bacteria and fungi will rapidly outcompete the plant tissue for those nutrients if they aren't eradicated. To achieve absolute sterility, laboratories use an autoclave, a heavy-duty industrial pressure chamber. The physical parameters are non-negotiable. The equipment and mediums are exposed to 121 degrees Celsius at 15 pounds per square inch of pressure for exactly 20 minutes. This destroys all microbial life. Absolute control over a sterile environment is the prerequisite for engineering biological replication. Without it, the process fails before it begins. With a sterile environment secured, we place the explant into the agar dish. The cells rapidly divide, forming an unorganized, undifferentiated mass of stem cells known as a callus. Over time, the callus begins to differentiate. It develops rudimentary roots and shoots, officially transitioning into what biologists call plantlets. These plantlets are carefully matured in test tubes until they harden enough to be transferred back out into a natural soil environment. This continuous cycle yields thousands of exact, pathogen-free clones of the original donor. Tissue culture completely bypasses the unpredictable timeline of germinating seeds. It provides the mechanism for rapid, industrial-scale multiplication of optimal genetics. Scaling identical traits is powerful, but modern biotechnology goes a step further. We can transfer entirely new traits across taxonomic boundaries through genetic engineering. Splicing genes requires two mandatory biological components: the target gene, which acts as the donor DNA, and a plasmid vector to transport it. A plasmid is a small, circular DNA molecule naturally found in bacteria, like E. coli. It replicates independently of chromosomal DNA, making it the ideal delivery vehicle. To insert the new trait, we have to cut the DNA. We use restriction enzymes, molecular scissors specifically programmed to identify and sever the DNA strands at exact nucleotide sequences. The enzyme physically binds tightly to the double helix, severing the chemical backbone to cleanly isolate the target gene. By isolating this exact sequence, scientists extract a functional biological code ready to be installed into a new operating system. Here is the architectural blueprint for assembly. We have the severed donor gene and an opened E. coli plasmid waiting to receive it. In the ligation phase, an enzyme called DNA ligase acts as a molecular glue. It fuses the chemical backbones of the target gene and the plasmid together. This fusion creates what is known as a recombinant DNA plasmid, a continuous transgenic loop of code. Next is the transformation process, where the host bacterial cell is forced to absorb this newly engineered recombinant plasmid. Finally, in the cloning phase, the bacteria reproduce exponentially. They turn into microscopic, self-replicating factories that continuously express the newly inserted trait. By splicing a single gene, we permanently reprogram a living organism to execute commands it never evolved to perform. Compare this recombinant technology against the historical baseline of selective breeding. For centuries, modifying crops or livestock meant relying on messy, unpredictable genome mixing. It was strictly limited to closely related species operating over long generational timelines. This chart illustrates the contrast. Genetic engineering drops the generational weight. It is surgically precise, immediate, and capable of crossing any taxonomic boundary, like moving a bacterial gene directly into a plant. We are no longer restricted to waiting for favorable evolutionary mutations. We possess the mechanics for deliberate biological intervention. These micromolecular tools scale up to solve massive real-world problems in health and agriculture. In agriculture, scientists have inserted Bt bacterial toxins into corn so the plant naturally resists pests, and fortified rice genomes with vitamin A to combat global malnutrition. In medicine, the exact E. coli plasmid process we just mapped out is used to force bacteria to synthesize pharmaceutical-grade human insulin. And in environmental protection, we can deploy specific engineered strains of Pseudomonas bacteria, explicitly designed to break down the hydrocarbon pollution choking oceans after an oil spill. But rewriting life carries extreme risk. Deploying genetically modified organisms demands strict adherence to internationally recognized biosafety guidelines to predict and mitigate unintended ecological impacts. The ability to program these outcomes offers a pathway to address global agricultural and medical challenges, provided we architect the code with scientific responsibility.